Hello. Today we're going to be discussing how eukaryotes control gene expression or turn genes on and off. So far we've been discussing how an operon system, which is how prokaryotes control their gene expression. We're now going to be looking at eukaryotes, and because eukaryotes have a nucleus and that the mRNA after it's transcribed needs to be transported to the ribosome, eukaryotes have many more ways to control their gene expression, or to turn genes on and turn genes off. So the big questions that we're going to be looking at, and then the first one really is our main sort of topic. We're going to be looking at 2 and 3, sort of the 2 weeks before Christmas, like after our test. We're going to be looking more deeply at those questions. But today we're going to be focusing on how genes in eukaryotes are turned on and off, how genes can be turned off if there's a changing condition. So if something changes in the cell, how can we turn genes on and off? And then how do cells during embryonic development, how do they differentiate into different cell types? And that's something we're definitely going to be looking at more after our test on all of the DNA stuff. If you look at the bottom, we see a growing human embryo, and you'll sort of see, in the beginning of the early embryo, it doesn't look like there's that many different cell types. But then, we start getting the development of fingers and feet. And so the question is, how does that happen? How do we turn genes on and off to allow different cell types to develop? Just to step aside for a second, in terms of gene expression, there's this whole new field in biology called epigenetics, which is the study of how sort of the structure of DNA, not the sequence of nucleotides, but how the structure of DNA affects its expression. So our scientists have discovered that it's not only the order of the A, T, G's, and C's, but how the structure of DNA can be modified to change its expression. And that's what we're going to look at briefly now. So if you look, there are 7 ways that you can control gene expression in eukaryotes. I'm not going to read them to you. The first 2 are pre-transcriptional. They occur before transcription occurs. 3, 4, 5, 6, and 7 are post-transcriptional, and they occur after transcription. In this video, we're going to look at 1 and 2, and then you'll get a second video that looks at the other 3, 4, 5, 6, and 7. But right now, we're going to be looking at how packing and unpacking of DNA controls transcription, and how we can either stop or start transcription in the first place. So DNA is really long. And in the nucleus, it's compacted. Okay? We are not going to go through all the levels of compaction. This was something that was on the old test. But essentially, DNA wraps around these things called histone proteins. And then these histone proteins come together. So if we take a look at the picture, these are the histone proteins, and the blue little line is the DNA. These histone proteins come together to sort of pack the DNA. When you have a DNA wrapped around a histone protein, there are 8 of them in this little circle, it's called a nucleosome. And then the nucleosomes compact together, and then the strands of nucleosomes pack together until you eventually get a chromosome. But DNA, if it was stretched, would not fit all the way out, would definitely not fit in your cell. It's really long, and it's compacted inside your nucleus. But the question is, how does this relate to turning genes on and turning genes off? But before we discuss that, let's look a little bit at nucleosomes and how they compact DNA. So nucleosomes are composed of the DNA that is there, and then the histone proteins, which are represented by these red, and the linker is the yellow protein here. Histone proteins are composed of 8 protein molecules. Okay? And they're mostly composed of positively charged amino acids, which is helpful because DNA, because of the phosphate group, is negatively charged, and therefore it's attracted to the histone proteins. And the DNA wraps around these histone proteins using this linker protein here, which the details of you don't need to know. And then these histone proteins, which the DNA wraps around, compact further to form a supercoil, which eventually forms a chromosome. Now, how this controls gene expression is that if DNA is tightly wrapped around histone proteins, so if it's tightly wrapped around the histone proteins, transcription can occur because RNA polymerase can't get into the DNA. And therefore, the genes are turned off. Just some vocabulary words. If DNA is said to be in heterochromatin form, it's tightly packed and not expressed. If DNA is said to be in the euchromatin form, it's loosely packed and can be expressed. If we look at the picture here, the dark sort of areas of DNA are heterochromatin, and the lighter areas are euchromatin. The question now is, how do we cause DNA to be loosely packed or not loosely packed? And we have an answer for that. So if you want to tightly pack the DNA, you methylate it. A methyl group is CH3. Let me see if I can write that. Point that will work. So a methyl group is it's right here. Oh, look at that. So a methyl group is CH3. And if you add CH3 or a methyl group to cysteine nucleotides, it causes the DNA to tightly pack, and that blocks both transcription factors, which we'll discuss in a second, and RNA polymerase from getting to the DNA. As a result, the gene is turned off. So methylation causes the DNA to tightly compact, and then as a result, turns genes off. In the reverse sort of way, if you want to unwind the DNA, you acetylate the DNA by adding an acetyl group. Okay? This causes the histone proteins to change conformation and causes the DNA to unwrap. Since the DNA is unwrapped, this allows RNA polymerase to get to the gene and transcribe it, and then as a result, the genes are turned on. This field of what we just sort of went through of epigenetics is growing. Like, if you can study epigenetics, you'll have a job immediately. But essentially, these are changes in the structure of the DNA, not the A, T, Gs, and Cs, that cause expression to be changed. And this has been linked to many diseases, and I've seen it's been linked to cancer, some of the changes that shouldn't occur or should occur. It's definitely used in development. But this is huge, a huge field that's developing. Like I said, this is very important in embryonic development. You do not want all genes turned on in all cells. Your blood cells, red blood cells, only need hemoglobin. As a result, many of the genes that you don't want turned on in cell types are heavily methylated so that they're not expressed and they're always tightly wrapped and will never be expressed in that cell. That happens during embryonic development. We're going to look a little bit like that after we have our test on this sort of general eukaryotic gene transcription. We're going to look how that's done during embryonic development. Now we're going to look at how transcription is started, or how you can start transcription if you want a gene turned on, or stop transcription if you want a gene turned off. There are more than just sort of the coding region of the DNA in terms of if you have parts of a gene. There are control sequences that control whether a gene is turned on and turned off. So in a gene, you have the control sequences, which we're going to look at in a second. So that's what I would write down here. And then you have the part of the DNA that codes for the protein. We're going to look at what these control sequences are. So if we look at sort of the parts of a gene, we have the coding region, which is what codes for the protein. And then we have the promoter. And at the promoter, there are two things that bind. We have transcription factors, which bind to the promoter, and their job is to bring RNA polymerase to the DNA. Okay? And they bind at the promoter. So transcription factors bind to the promoter, and they bring RNA polymerase to the DNA. Then upstream from the promoter in the coding region, we have enhancer sequences, which are also sometimes called switches. And what's required to sometimes turn a gene on is other activators, which are also known as transcription factors, but usually they're transcriptional activators, to bind to these enhancer sequences. Usually what happens, this causes when the activator binds here, it causes the DNA to bend. And that allows RNA polymerase to come to the gene and transcribe it. So now we're going to look at something that's really important, and that I've talked about a lot, but never sort of formally defined or explained what it was. We talked about transcription factors when we were talking about signal transduction pathways, and how sometimes the response is a transcription factor going into the nucleus and turning on a gene, which makes a protein. We're going to look a little bit more deeply about what transcription factors are. So transcription factors are protein molecules that turn genes on most of the time. There are some negative transcription factors that can also turn genes off. There are two types of transcription factors. The first type is called basal transcription factors, and these are required for all genes to be turned on. Like every single gene requires the same sort of basal transcription factors to bind to the promoter so RNA polymerase can bind. Then there are regulatory transcription factors, and these are specific for a specific gene. They recognize and bind to specific sequences in the DNA, usually at the enhancer region, which you remember is upstream from the DNA, usually at the enhancer region, and they either turn on, if they're activators, can turn on the gene, or repressors that could turn off the gene. But often, these activators are required to start transcription from many genes, and they bind to specific sequences in the DNA, and they're specific for specific genes. So if we look at here the sort of transcriptional unit that's needed to turn on transcription, in green we have the basal transcription factors, which bind to the TATA box in the promoter, and they are required for all genes to be transcribed. These need to be here for any gene to be transcribed. They specifically bind to the TATA box, and that's the names you don't need to know or memorize, but they are required for all genes to be turned on. Then if you look, we have these sort of red and brown activators. These are specific transcription factors, and they bind to enhancer regions or the switches. And what they do is they cause the DNA to bend. Okay, this bending of the DNA, along with the presence of the basal transcription factors, allows RNA polymerase to come to the gene and transcribe it. If these activators are not here, then the gene can't be transcribed because RNA polymerase will not be brought to the gene. Remember, though, these activators are specific for specific genes and bind to specific regions based on the sequence of DNA. So these enhancer regions are DNA sequences. They're A, T, Gs, and Cs. And their sequence is specific so that this protein can bind to it and recognize that sequence, and only that sequence. There are also repressor proteins, they're not really called repressor proteins. Usually they're called silencers, but different from the repressor protein in the operon. And what they do is they bind to the enhancers as well, but they block activators from binding. So then as a result, they can't bind, and then RNA polymerase can't be brought to the DNA. Here we sort of have the same picture, just sort of in a more simplified component. Again, what I would do, I'm not going to do this for you, is I would label what these things do. The only thing that's new here is the mediators. And mediators allow activators or transcriptional activators to sort of bind to the general transcription factors to allow the DNA to bend. But I would make sure that you label what all these do. Remember, activators or transcription factors, they're just specific transcription factors that bind to enhancers or switches, those are the same word, to either turn on genes if they're activators, or turn off genes if they're repressors. But remember, they are specific for specific genes. Just to bring back an example to relate this to old stuff. If you remember when we talked about cell cycle control, we wanted to give the cell a signal to divide because of some changing factor in the cell or damage to skin cells or whatever. A growth factor, a ligand, bound to a receptor or molecule causing a conformational change. This then activated a protein called RAS, which then activated another protein called RAF through phosphorylation, and then RAF activated another protein through phosphorylation, which then activated another protein through phosphorylation. The final protein that was activated, if you remember, was a transcription factor. And remember, the transcription factors now, we know, turn on genes and cells. The transcription factor, what the transcription factor did though, which we didn't talk about then, is the transcription factor here, that's like activated at the end, binds the promoter. Okay, so this transcription factor that's now here, we're zooming in on here, binds to the promoter region, which allows RNA polymerase to come to the gene and transcribe this segment of DNA. If you remember, what it's transcribing in this case is cyclin. And then cyclin gets transcribed and translated and turned into the protein cyclin, which can then bind and activate CDK. So that idea of how the cell responds using a transcription factor is now a little bit more obvious. All right, do the guided questions, and I'll see you guys tomorrow, Thursday, Ep School.